Okay.
Thank you, Mackie, and thank you everyone for attending. So Mackie said that I was a practitioner for years, and I loved it. And actually, when I left New York, I had all intentions of going back to being a practitioner. I never really saw myself as an academic. And then an opportunity presented itself after I got my PhD to work at Tennessee, where they have the body farm. It's actually called the Forensic Anthropology Center, or the Anthropological Research Center. And I saw it as an opportunity to do research related to practical questions. So questions that I had as a practitioner, things I didn't quite understand that I wanted to do research on. And so I took a chance, thinking, if I don't like it, I'll leave. That was 11 years ago. I love it. I love teaching. I love doing research. I have phenomenal graduate students and undergrad students, and I get to teach fun classes. So I'm super happy here. And I get to do research that is related to the work I did as a practitioner, to help answer questions we had when we were doing casework. So I'm just going to give you a little introduction to the body farm, which we now have finally given in, and we no longer correct people when they call it the body farm. We even call it the body farm. I think our Facebook page is the body farm. But, we do more often than not refer to it as the forensic facility, out of respect to the donors and the donors' families. We couldn't do any of the research that we do without people donating their bodies to us for research. So just like you would donate to a medical school, people donate their bodies to us. And so we have to be very respectful of those individuals and their families. And interestingly, family members of donors stay very involved with our facility. Once a year, we have an open house for family members to come and learn about the research that's being done using the skeletal remains of their family members. They are allowed to see the skeletal remains if they want to, because they don't have a burial plot or a cemetery or somewhere to go where their loved one may be buried. So we stay very involved, and families want to know that the remains are being used for research. In fact, we changed the way we stored our skeletons because we found that skeletons that were on the highest shelf, that were hard to reach, often weren't used for research because people couldn't get them down. And families would want to know, why wasn't my loved one used in many studies last year? And it turned out that a lot of those were too hard to reach. So, so we're very invested and close with the families that we work with who donate their loved ones for us for research. So I'm going to talk to you about some of the research that goes on at the facility. Most of what I'll talk about are projects that I am involved in, either as the PI or tangentially, although I will mention some other projects that are going on that I think are interesting and just kind of give you an overview of the type of work that's done at the facility. So the facility was formally established in 1987 by William Bass, although he did receive his first body donation in 1980. Initially, it started with a very small parcel of land, but we've since gotten additional bits of land that we've added on. They're adjoining, and we now have about 3 acres. We have a 5,000 square foot building that has a classroom that we use for teaching. We do short courses there. We have, we do FBI courses there. We do university courses, summer courses. We also have a bay, which is like an intake bay, like you would see at a mortuary, where the bodies come in and we do all the intake work. So we collect hair, muscle, photos, blood. We do height and weight and all that before the body is placed out at the facility. The building also has all of the room for maceration. So if bodies don't fully decompose, when we collect the skeletonized remains, we can clean them up a little bit and accession them. We did have room for casework, but we've gotten so busy that they're now building a new building just for casework. So just for forensic casework, because there are a number of forensic anthropologists in our program who do casework, and that new building will go through the accreditation process. Typically, we have about 150 to 200 donors out at the facility at any given time. Most of them are enrolled in different research projects. And I say typically because everyone knows there's nothing typical about this year. When COVID started, we shut down and we stopped accepting bodies because no one really knew how COVID would transmit postmortem, but also because we didn't want to use up PPE. We have slowly opened again and now accept bodies, but only bodies that the medical examiner has tested and they've tested negative for COVID. We don't accept any bodies with certain communicable diseases. So no hepatitis, no HIV, no MRSA, anything that may put our team members at risk. So the body donation program actually has thousands of pre-registered donors. So people like you and me who are alive, who fill out the paperwork and say, I want to donate my body when I die. And a lot of those individuals actually come to the BASS building to participate in ante-mortem research, like biometric research. So we'll collect iris scans and fingerprint scans and face scans. And then when they die, we continue that biometric work to see how those processes work with decomposing remains. So that's some of the research that people at the facility are involved in. In fact, we have so many pre-donors that we pretty much only accept people who are pre-registered. So when you're pre-registered, you fill out a form. Where were you born? Where are all the places that you lived? What was your job? Height, weight, medical records, medications, surgeries. We collect all kinds of demographic data because a lot of that is used later in our research. We've done research on individuals with breast cancer, with prostate cancer, looking at their radiographs. And so we need to have all that health history and life history for like isotope studies. When we have someone who's not a pre-donor, we often don't have that information. And while we don't like to turn people away, that information is really important for some of the research that's being done. We now have the largest collection of contemporary skeletons in the US, so modern skeletons, and there are a lot of collections of skeletons. Smithsonian has some, different universities do, but ours is modern, and we see changes in the skeleton that happen over time. And so these skeletons are used for current forensic research. We have more than 1,800 individuals in our collection now. We also have a number of cremations. When someone wants to donate a cremation, we just ask that it's not pulverized. Because after someone is cremated, the burned remains are then pulverized. But before that, there are actually quite a number of recognizable fragments in a cremation. And we have a number of fetal remains as well. So the mission of the Forensic Anthropology Center is to achieve excellence in research, training, and service in forensic anthropology and related fields. And when I say related fields, it's important because we rarely work in isolation. Almost all the projects that happen at the facility are multidisciplinary. I've worked with soil scientists, plant scientists, engineers, molecular biologists from a number of different agencies. You know, everything we do is multidisciplinary. This is a long list and it's not comprehensive, but these are some of the areas of research that people are studying at the facility. Entomology, we have a postdoc now who's an entomologist. Trauma studies, we have two individuals who have a National Institute of Justice grant looking at sharp force trauma and how it's affected from burning. So they've been working with the engineering department. They built a machine to simulate sharp force trauma, and then off-site, we have an area where they have a pyre and actually burn the individuals and then look at the changes from burning in the sharp force trauma. Post-mortem interval studies, how long someone's been dead. Taphonomy, what happens to the body after death. Mummifications, scavenging, it's a big deal. Microbial studies are huge now. Everyone wants to study microbes of dead bodies. Biometrics, forensic documentation. When you're documenting a crime scene, is it better to use photogrammetry or 3D scanning? We've done comparisons during excavations of graves. DNA studies, isotope studies, changes in the soil, changes in the plants, remote sensing, metabolomics, proteomics, animal proxy studies, a little bit of everything. If you can think of it, we'll do it. So one of the early studies I was involved in was animals as proxies. And the reason that we did this is because there are lots of publications that say pigs are great proxies for humans. Except if you think about a pig, the shape of their body is really not the same as ours. Like they have these little stubby limbs. We have like long skinny limbs. They have very thick skin. Our diet, while they're omnivores, is different. So we did a number of studies where we had a pig, a rabbit, and a human. We placed 5 of each, and we did this 5 separate times during different seasons, captured a ton of information, and determined that non-human substitutes cannot capture the range of variation that manifests during decomposition. So what do I mean by that? Well, first of all, rabbits, forget it. There was nothing similar about humans and rabbits. So people need to stop using rabbits. Interestingly, scavengers, and we have a lot of raccoons, and we put game cameras out there and we watch them. They come out at night and they bring their little raccoon babies with them, and they scavenge, eat remains, and they like humans way more than pig. Maybe it's our high sugar diet. We have noticed anecdotally differences in decomposition between individuals who have diabetes and people who don't. So clearly there's something in our metabolism, in the way that we smell or taste that's different. Scavengers didn't touch the pigs, they didn't love the humans. Conversely, the maggots quickly colonize the pig before the human and decompose the pig way faster than the human. And really interestingly, neither the pig nor the rabbit mummified, but the humans mummified a lot. So there were enough differences that animals don't make good proxy studies if that's the end of the line. And I say if that's the end of the line because we do ask people to do animal studies first before human studies as a proof of concept, if it's something new. So if it's a new, especially if it's destructive, we want to know that it works on animal tissue or animal bone before we move to human, because once we start destroying human tissue, we can't get it back. Some interesting biometric studies going on. Current sensors that are out there are used on living individuals. But when a person dies, there are changes that happen. Their vitreous fluid, even the way that they hold their face, the muscles, things change. And whether those biometric tools still work on deceased individuals and for how long, we're not sure. So we have a number of individuals who've been working on biometric studies, looking at face, iris, and fingerprint for the most part. Life history from isotopes. An application of life history approaches to stable isotope analysis uses different tissues, bone, tooth, muscle, that gives us a snapshot of where that person was from. And that's because these tissues record elements from the environment. So oxygen gets recorded in the bone. When you drink water, different elements from the water get recorded. And when we look at those isotopes in an individual, it gives us a snapshot of what they were exposed to while those tissues were developing. So if we want to know about where someone lived and grew up when they were a child, we look at their teeth, because your teeth developed, the enamel in your teeth developed when you were a child, and it doesn't change throughout your life. Whereas your bones remodel continuously. They're a living tissue. And so bones remodel over about 10 years. So if you want to know where someone lived in the last 10 years or so, you look at their bones. If you want a snapshot of the last few months in their life, you can look at their hair and their fingernails, because that grows at a much faster rate. Now, does this give you an address? No. Does it give you a state? Not usually. It'll give you a region. So this is often used on individuals, say, for example, a body has been found along the border. It may tell us from what region that individual has traveled from. And these are just some of the different isotopes that individuals have looked at. And again, hair, nails, bone, and teeth. So this is a study that I did with some colleagues at the International Commission of Missing Persons. So as Becky said, we had worked on the World Trade Center, and we worked on some plane crashes. And I'd worked on a number of other disasters. I was talking to the folks at the ICMP. There was that's the International Commission of Missing Persons. They're the individuals who led the identifications of the victims from the former Yugoslavia. And we were talking about how following a disaster, there's this expectation in Western countries or countries that have enough money that you're gonna have an ID and it's gonna be done by DNA. And then there's this assumption in countries that can't afford it, that too bad, then you don't get it. And that's just not acceptable. And so we were trying to find a new method of collecting postmortem DNA that was easy to collect, you could collect it quickly, that was stable. So if you're in a country post-disaster where there's no electricity and there's no refrigeration, and there aren't a ton of forensically trained people to collect the samples, we could still preserve something, even if we had to do mass burials, that could later be used for identification. So we decided to go back to the basics. FTA cards, they're used for paternity testing, right? I mean, most labs can test an FTA card. Not every lab can test bone, but they can test an FTA card. And so we tried this method where we made an incision, we used a swab, we rubbed it on an FTA card, a change in color one, so we knew that a sample had been collected. The nice thing about an FTA card, you leave it out for 10 seconds and it dries. You close it. It doesn't have to be refrigerated. It's not soft tissue that's decomposing. It's not bone that has to be taken out with a bone saw. It doesn't have to be stored anywhere special. It could sit on the counter. And we tested individuals over, I want to say 6 weeks, 65 days. In the winter, in the summer, in the fall, in the spring, all different time periods, to see how long this method lasted. When do we have to move to bone? Because bone makes it more challenging. Bone's more expensive, it's harder to process, it's harder to sample. And we correlated this data with a method that we use in anthropology to estimate postmortem interval. It's called the total body score. So we look at a body, look at different parts of it, the head, the torso, the arms, the legs, and we can score it based on its level of decomposition. And what we found, and this is just a few bits of the data, it's not the whole thing. What we found was it didn't matter the season, winter, summer, if the total body score was under 15, we got a full profile. If it was over 15, it started to precipitously drop. And so you could reach 15 in 5 days in the summer. You could not reach 15 in the winter in 2 months. So it's not a season thing. And it's so interesting because so often someone would say, well, how long can the body sit out and you can still get DNA? But how long is irrelevant. How long is, it's based on context. So how long in the summer in Tennessee or how long in the winter in Maine is very different. And so really what we came up with is if you do the total body score and the individual is under a certain score, chances are the tissue is still going to work. And just to be clear, I may play a DNA scientist on TV, but I am not. I'm not a DNA person. I understand it. I work with a lot of them, but the folks at ICMP did all the DNA work on this. I did the body scores and the sampling. I've also been involved with some skeletal DNA, and this stemmed directly from the World Trade Center. One of the things that we noticed in the identifications, so remember, this is back in early 2000 when DNA from bone was still pretty boutique work. It wasn't done on a mass scale. And all of the manuals said, weight-bearing bone. And we kept seeing little foot bones, little hand bones, random bones that had never really been tested before, yielding lovely full profiles. So when I got here, I started working with the folks at Ph.D. Technology, who did all the DNA from the World Trade Center. And we decided to do a prospective study. We looked at 3 individuals and we tested every bone in their body. And we found that some of the small cancellous bones outperformed the weight-bearing cortical bones. So we thought, okay, well, these individuals are pretty fresh. Bone is hematopoietic, that's where we make our blood. Maybe it's different if they're older skeletal remains, if they're old and dry bones. So we took the 10 best yielding bones, and we took 12 more skeletons from increasing postmortem intervals out to 20 years. And we found that the order maintained. So we thought, okay, well, these individuals are on the surface. They're all decomposed on the surface. Maybe it's different if they're buried. You know, once they're buried, the microbes, there are a lot more microbes in the soil than on the surface. So I had a doctoral student, and this was her dissertation, and she decided to add microbes in because that was her jam. She's now doing a postdoc in a microbe lab. So first she wanted to know, are there differences in patterns of preservation of skeletal DNA between surface and buried? So we had 3 individuals that had been buried for 4 years that we dug up, and she did a comparison, same exact methods, same bones. And then she wanted to know, is susceptibility to microbial invasion related to DNA preservation? So she worked very closely with the folks in soil science who specialize in soil microbes, doing some 16S and 18S studies of the microbes that co-isolated with the DNA. Again, she found that bones of the feet performed the best, and it was consistent across the surface and the burials, and that total bacterial and fungal gene abundances were not reliably predictors of human DNA concentration, which we actually did not expect. We kind of thought it would be a good predictor. She then went on to ask what microbes can colonize bone, and does it differ by anatomical region? And so she compared the surface to the buried, but then she also used the American Gut Project to get a good understanding of the microbes that were most associated with the gut, to see, were we seeing differences in regions? Were gut microbes colonizing bone closest to the gut? She found that a number of diverse phyla are present in bone, and that some of them are likely to colonize bone and impact skeletal degradation, and that they do cluster by body site, especially the feet. They really clustered by body site. And then she was looking at environment, surface versus subsurface. And water was a major factor between the 2 environments, because there was a water table on the burial that would periodically rise. And so there were some microbes associated with a more wet environment that were found in the body at the bottom of the grave compared to the top. Okay, just keep track of time. So this project is very close to my heart. This is one I've been working on for years. My graduate advisor, my doctoral advisor, was the director of forensic science at the International Commission of Missing Persons before Tom Parsons. And I went to visit him in Bosnia, and he was telling me that, you know, there were still thousands of individuals missing, and they had excavated all the graves that they knew about, all the witness testimony, everything that they had information on. They just didn't know where the rest were. So we started thinking about it, and when I got to Tennessee, I decided to work with some folks in remote sensing to see if we could find other ways to locate clandestine graves. So we got a parcel of land that was previously unused because we wanted to leverage the signatures of decomposition. And if we used land that had already been used, they would already have decomposition all over it. So we used a new parcel of land. We buried a single individual in a grave. We buried 3 individuals in a grave. We buried 6 individuals in a grave. And then we buried 24 individuals in a grave. And we also put in some control graves because there's probably a signature from disturbed soil that is different from disturbed soil with a body decomposing in it compared to undisturbed soil. And then we waited. And then we begged lots of agencies for money. And we begged lots of agencies to help us. And we got some good donations because we're anthropology. We are probably one of the poorest fields out there. And we collected ground, air, and space data. So on the ground, that was our ground truth. The air data we collected, we contracted with some folks at NASA who flew an airplane about 500 feet above our site and collected data from the plane. And then we also got some donated satellite data. So this is what the site looks like. And as you can see in the slide beforehand, we did this all by hand, volunteers, grad students, and faculty. We were all out there digging, and that red means clay, so it was not easy. This is the 6th person. This is the exact same size, no bodies, so that was a control. And this picture was taken on February 15th. Yes, we interred everyone on Valentine's Day. This is what it looked like on the day after we closed everything up. 2.5 months later, this is what the site looked like. So you can understand why graves are hard to find once the vegetation is taken over. I mean, obviously, if you look at the first slide, you know, I know where that is. I know where the bodies are. I can see where they dug the hole. Come April, vegetation is totally covering it. Take yourself to 1 year later, and the earth has totally healed up. The scars are gone. And so when you fly over an area and you do aerial photography, if there's a recent scar, you know where the bodies are. But once that scar is gone, then it becomes very challenging to find those bodies. So this was after 1 year. This was the 24 person after 2 months. This is the 24 person after 12 months. 18 months, 21 months. One of the things we looked at were temperature trends. So first we used these little things called the eye buttons. They look like nickels. We put them in the ground and they record temperature changes. Because if you think about it, when a body decomposes, it generates a lot of heat. And we were able to have an understanding of when decomposition was happening because we could record that information. For the 24-person grave and for the 24-person control, we actually used a distributed temperature sensor, so a fiber optics cable that we put in below the bodies, between the bodies, and above the body to capture real-time temperature. We also had dedicated FLIR cameras, which captured thermal imaging to see if we could see some of the thermal changes. On the ground, and this is another doctoral student, Katie Corcoran. This was her dissertation. On the ground, we collected spectroscopy and LIDAR. So for the spectroscopy, we use an ASD field spect, which captures high resolution spectra from the vegetation. And what it's looking at are the chemical changes in the vegetation, because when vegetation is stressed, it changes its signature. You know, it may accept more chloroform or not. And so when a body decomposes, we're full of nitrogen. I mean, we're at the top of the food chart in nitrogen. You know, if you think about like plants are here, animals that eat plants are here, animals that eat animals are here, and humans are here. So when humans decompose in the vegetation, all of a sudden the vegetation is flooded with all this extra nitrogen, and it stresses out the vegetation, and that can be picked up spectrally. So we were trying to leverage decomposition to see these changes. We were also using LIDAR, which is, it collects information on elevation changes, but it collects it at a very, very, very micro scale. So when you think about the earth, when you look at a field, there's these natural undulations all over the ground. And this will measure those within the centimeter. When you have a burial, when you take all that dirt out, you put a body in, that body displaces some space. You put the dirt back in, you're gonna have a mound because you can't get it all back in. But it's loose. It used to be really packed tight, really compact dirt. But when you dig it up, you aerate it and it's loose. Put it back in, and then you've got gravity and you've got rain. And shortly after, you see a drop in elevation. And that drop is the same shape as the grave because everything starts to compact. Whether there's body in there or not, you see that drop because it's compacting. The one without the body stays like that. The one with the body, once the body decomposes, all of the space that the soft tissue took up is now gone. And the soil starts to backfill inside the skeleton, and you see a second drop. So you see another little divot in the grave. And we can measure that. We can measure when it happens and how far it drops. And here's an example of what it looks like. So the white is above ground. So right here, the dark is deep. This is the 6-person grave, empty. This is the back dirt pile. So we have a big pile of dirt here and a big hole here. On day 108, you can see where it is here, the shape of the grave. It has dropped about 0.6 centimeters. When we look at day 40 something, it's blocked on my screen, you can still see the shape of the grave, but there's one area where it's dropped even further. And interestingly, this grave is 2 meters by 4 meters, but all the bodies are on one end. Which end do you think the bodies are in? The green one. That's where we see a even further drop. That's that second drop. So this study was super interesting, but had a ton of limitations. So we're redoing it. We did find that you can easily separate the vegetation spectrally from disturbed and undisturbed. We also collected vegetation growing over the graves and found that there were significant differences in the isotope levels, the nitrogen isotope levels in the vegetation growing on and off the grave. We saw observable elevation changes that were spatially clustered. So we could see where the graves were. They looked different from the natural undulations in the earth. But our resolution wasn't good enough in terms of temporal resolution because we didn't collect as often as we had anticipated because of some limitations with the equipment, which we now own. So we had some snapshot time points. We would like to have more regular data collections. There's a lot of limitations when it comes to canopy. LIDAR can capture information below canopy. It doesn't actually penetrate canopy, but because it shoots down so many laser points, eventually some get through, but not as high resolution as we had hoped. Some of our spatial resolution from our aerial data wasn't as good as we hoped. It was about 1 meter. So if your grave is 1 meter by 2 meter, you could miss it because it's only showing up as 1 pixel. But again, this was almost 10 years ago when we started this project. So now resolution is much better. We'd like to do some more correlation studies between the spectra and the isotopes, because the spectra is picking up the chemical changes, the isotopes are chemical changes, and they should correlate with each other. We've actually just started that project. And ultimately, the goal for this, instead of having, you know, a $200,000 piece of equipment here, an $80,000 piece of equipment here to collect this data, is to find the specific signatures so we can use less expensive equipment and put it on a UAV and be able to collect data in places that are not necessarily the safest for forensic practitioners to go into, to either locate or remotely monitor graves until it's safe enough to go in there and do the work. And then our whole study was specific to East Tennessee. Who knows if this would work in California, Texas, Vietnam, Bosnia. You know, the vegetation is different, the soils are different. So more replicate, I mean, you're scientists, more replicate studies. Keep repeating it, using different parameters. But that's the gist of it. So, any questions?
Yeah, thank you. I was just about to give you a time warning, but if you're ready for questions now,
yeah,
that would be great. So please, everybody can you can just unmute yourself to ask your question.